So I'm going to talk to you about a very specific mutation that's been found in acute myeloid leukemia and why we think that this mutation is important. So we know that acute myeloid leukemia is a very deadly disease, and unfortunately, apart from some exceptional subtypes, the prognosis hasn't really changed over the last 30 years. And also the treatment has stayed relatively the same. It's standard chemotherapy, the so-called 3+7 regimen. And although the majority of patients actually respond to the chemotherapy pretty well initially, unfortunately, most of them will later relapse because they develop, and this relapse disease is going to be refractory to the chemotherapy. And it's believed that oftentimes it happens because there is a small subset of cells, of leukemic cells, that are resistant to the initial chemotherapy, and they'll just stay in the body and persist there, and they would later give rise to this relapse disease. And so those cells are called the so-called minimal residual disease, and they're believed to be chemoresistant pre-leukemic stem cells. And we wanted to investigate whether there was a link between specific mutations that are found in leukemia, presence of the minimal residual disease, and the mechanisms, the specific mechanisms of chemoresistance. So we performed a very large analysis on a large clinical trial that has been done in the United States. It's called an ECOG, an Eastern Collaborative Oncology Group E1900 clinical trial that's actually been reported a couple years ago. And it involved 398 AML patients. And we correlated the presence of minimal residual disease that we can detect by some very sensitive methods, by flow cytometry, where we take the blood out and bone marrow out, and by using specific, very sensitive method, try to detect the disease. So we tried to correlate the presence of minimal residual disease and specific mutations that were found. Later, we also wanted to see how these mutations that we found, what was the mechanism of driving chemoresistant disease by using a mouse model. So moving fully into the basic science department and trying to model what's going on in patients. And some of the results included that there was only one particular mutation that robustly correlated with the presence of minimal residual disease. And it was a mutation in a gene called DNMT3A. And it's not just any mutation in this gene. There is one very specific mutation that just affects one point in this gene. And the presence of this mutation predicted both the presence of minimal residual disease and the presence of chemotherapy and adverse outcome. And so we took this mutation and put it into a mouse to see what's going to happen. And actually, just this mutation alone did not induce any disease in a mouse. However, when we combine it with some other mutations that are very commonly present in disease, we can see that the leukemia that we got in a mouse was much worse, was much more aggressive than the other mutations alone. And actually, the presence of this mutation is very interesting because last year, a couple of studies also identified this particular mutation in a large number of elderly people without any hematologic malignancies. But those people had so-called clonal hematopoiesis. It means that their blood in the entire body was produced pretty much by one stem cell that had this mutation. So it means that actually this particular mutation, maybe it doesn't cause leukemia, but it makes a really good stem cells that can outcompete all the other stem cells in the body. And one of the reasons why these stem cells are so good, because they're actually resistant to stresses, and chemotherapy is also a stress, so they're resistant to that stress. And so when we try to look at the mechanism on the molecular level, what we found, so when we treat cells with chemotherapy, usually we induce DNA damage. It means that there are breaks in the DNA. And in order to repair these breaks, the cell first needs to get access to those breaks. And DNA is not just sitting in the cell. It's actually spooled on little proteins that are called histones. And so the cells need to unspool the DNA first to get access to the break. And there are specific proteins that are involved in that. So what we found that DNMT3A actually inhibits this unspooling process. And it means that because this process is inhibited, the breaks persist because the cell just doesn't know that they're there and is unable to repair them. And as a result, the cell is unable to repair these breaks, and the cells accumulate mutations. And also, those cells are damaged, but they are not eliminated by apoptosis, as we heard earlier today from Dr. Roberts. So the cells are not being told to drop dead. So they're pretty bad, but they still survive because they don't know that they need to drop dead. And so we believe that this is the mechanism how the damaged cells survive low-dose chemotherapy. And this is actually also the mechanism how they accumulate additional mutations to accelerate the leukemogenesis later on. So the conclusions from our studies is that we believe that we found a novel mechanism of resistance to chemotherapy. And this is a novel mechanism that leads to chemoresistance and to persistence of this minimal residual disease that later gives rise to the relapse. And so this very specific mechanism, this defect in DNA damage repair, means that actually maybe we can find a way to target this specific vulnerability. Because in the same clinical trial earlier on, it was known that these patients who were resistant to standard dose chemotherapy, they actually benefited from the increased dose of chemotherapy, while all the other patients without this mutation did not see any benefit. They also saw an increased toxicity, but there was no benefit. So now we actually have a mechanistic basis to know why increased dose of chemotherapy in this particular group of patients may be good. And we can also find novel ways how to target that. Maybe we can try to target other components of the DNA damage machinery. So if we just hit the cells harder, they won't be able to survive. Or another avenue is actually combining chemotherapy with ABT199, again, as we heard from Dr. Roberts, also to promote apoptosis of those cells, so actually force them to drop dead. And so we're now investigating these multiple therapeutic avenues.